Tammany and everyone for allowing me to come and present to you today. And I'm presenting on behalf of Duke University, University of Southampton and University College London. And this is work related to my PhD. And the work that I'm going to present in particular is related to microcirculatory response to hypobaric hypoxia. So I'm a critical care nurse. As I said, I'm a PhD student. My day job is that I'm a senior research manager at University Hospital Southampton, working within the critical care research area with Mike Grocott and Denny Levitt, who you may have heard of in relation to perioperative medicine, possibly. And I'm also a member of the Extreme Everest executive team, two other members of whom you may recognise, Monty Mython and Dan Martin, and this is us just before we were going off to Everest in 2013. So my overall PhD has been looking at epigenetic markers of adaptation to hypoxia and related phenotypes, and it's been using a chamber model. And so the reason that we're taking people to simulated altitude is because when you increase the altitude, which you can see on the left of this screen, you get a corresponding reduction in barometric pressure and therefore a corresponding reduction in the available oxygen. And so if you think that at sea level you've got 21% of oxygen available, when you get up to about 3,400, which is the maximum altitude that we simulated in this study, the actual oxygen you have available to breathe is about 14%. So the reason that I'm interested in this is because of critical illness, and as we know, hypoxemia is common in critical illness. But Abdel Salam in 2006 reminded us that we need to be careful when we talk about hypoxemia and understand that it is different from tissue hypoxia, and that you can have a patient who is demonstrating normal oxygen levels in the tissue, but actually appear to be hypoxemic and vice versa. And we know that a response to hypoxia or hypoxemia is potentially a factor in clinical outcomes, that there's great heterogeneity between individuals, and the hope is that if we understand the physiology of response, we might improve survival. Now, as I said, the overall study that I've been doing is related to epigenetics, and just a bit of a review of what epigenetics is. It's the study of changes that affects the transcription and translation of the DNA, but it doesn't necessarily affect the underlying sequence. And these epigenetic changes that we most commonly look at are histone modification and DNA methylation, and these changes can be heritable and they can be reversible. So DNA, as I'm sure you'll remember, is composed of 4 nucleotides. You have a sugar, then you have one of 4 bases, which may be adenine, guanine, cytosine, or thymine, and then you have a phosphate. And you get methylation, which can occur on the cytosines that are located next to guanine nucleotides. And these are referred to as CPG sites, where the C relates to the cytosine, the G to the guanine, and P to the phosphate bond between the two. And you often get CPG islands, and these are regions that are rich, regions that are rich with these CPGs. And they are often located in the promoter regions of genes. And when you get methylation, it tends to repress the transcription of the DNA code. So we talk about it silencing genes. Glad to see I'm seeing lots of nods, Matt. I'm obviously saying something right. So if we look at this again, you can see that we have chromatin, which is the DNA here, and it tends to get wrapped into these nucleosomes. And so you can get histone modifications where you get acetylation of the histone tail, which is attached to the nucleosome, or you can get a methyl group attached to the nucleosome as well. And when a gene is switched on, you have no acetylation of these histone tails, and you have no methyl groups present, and so transcription is possible. But when you switch off the gene, that may be because you've got this methylation, these red blobs here, and you see that there's no acetylation of the histone tails. But I'm actually going to be concentrating in this talk on the microcirculation, which is one of the phenotypes that we looked at in relation to the individuals that we captured epigenetic data on. And the microcirculation is really the arterioles, venules and capillaries that are less than 100 microns in diameter, and they control localized blood flow. And there are a number of ways that you can actually measure the microcirculation. So you can use laser Doppler, which is what you see this kit on the left. You can use incident or side stream dark field cameras, or you can use near infrared spectroscopy. Now, we're interested in the microcirculation in critical illness, but again, in the same way that you need to define hypoxia as different from hypoxemia, we need to remember that the microcirculation is different from the macrocirculation. So you can have situations where the macrocirculation can look absolutely fine, but you've actually got tissue hypoxia. And Inja in 2015 described situations where you could resuscitate in sepsis to get good macrocirculation, but you would still have microcirculatory dysfunction. And this could relate to heterogeneic flow, reduction in capillary density, vasoconstriction, or tissue edema. And we've used, in our group, we've actually looked at the microcirculation in high altitude on a number of occasions. So Davis et al. In 2018 reported preserved microcirculatory perfusion in Sherpas versus lowlanders. Gilbert and Kawai in 2016 demonstrated in lowlanders that there was an increase in capillary density as individuals went to altitude and remained at altitude for long periods. And Martin et al. In 2010 demonstrated a reduction in microcirculatory flow index in lowlanders. And in this study in particular, this is one of the graphs from that study from Martin, you can see that we had two groups. So we had climbers who were going to the top of Everest, and then a base camp team who were stayed at Everest Base Camp at 5,300 meters. And as they first went up from baseline to 5,300 meters, there wasn't a lot of difference in the microcirculatory flow index, but as the group separated and the climbers went higher on the mountain, their microcirculatory flow index reduced significantly compared to the those that stayed at 5,300 meters. So the technique that we used in the study that I carried out was laser Doppler flowmetry, which I mentioned before, and this is a non-invasive technique, and it uses two probes. One is a laser probe, which uses a flux signal, and this is measured in perfusion units, and it relates to the speed and the concentration of the red blood cells. And then there's a second probe, which uses white light reflectron spectroscopy, and this gives us a measure of tissue hemoglobin and saturation. And both of the probes have thermistors in them, so you can measure local skin temperature throughout the study. So laser Doppler has been used in clinical studies. So there's reported assessment of microvascular dysfunction using laser Doppler in diabetes. So Human et al. Reported on a study in 2017. Krokowski looked at it in relation to measuring drug delivery using skin drug delivery, so looking at vasodilation. It's been used to look at determining patients' CHD score, which is a measure of cardiovascular dysfunction, and it's also been used to assess diabetes-related complications and see whether you can differentiate between individuals. So as I said, the overall PhD study has been looking at whether altitude naive males would exhibit physiological and epigenetic profiles distinct from altitude experienced males. And the reason for doing this is that we believe that individuals who've had previous hypoxic exposure demonstrate a difference in their profiles. And this is a study from Edwards in 2010 that we carried out in 2007. And this was looking at muscle phosphocreatine. So we were using magnetic resonance studies to look at phosphocreatine levels in these individuals. And the climbers had had quite a lot of previous hypoxic exposure, but the trekkers hadn't. And you can see even at baseline that there's a difference in levels of phosphocreatine. And the question is this because they were just different genetically, or is this that the climbers had had a previous hypoxic exposure, which meant that they had developed an increase in their phosphocreatine levels? So this study was carried out, as I said, at Duke University in their hyperbaric chamber. We did 2 separate chamber runs, so we could, because you can only fit a maximum of 6 individuals in this chamber at a time. So each chamber run had a mix of 3 altitude experienced and 3 altitude naive individuals. And the way that we determined whether they were altitude experienced or altitude naive is that altitude naive had not had to have had any previous ascent above 2,500 meters in their previous 5 years. And those who were altitude experienced had to have had at least 3 stays at more than 5,300 meters, which is the height of Everest Base Camp, on for about 3 weeks in the previous 3 years. So quite a significant hypoxic hit. And so what we did was we carried out some baseline testing, and then we put them, sealed them in the chamber for 4 days and took them up to the equivalent of 3,500 meters, and did at baseline and then during the ascent repeat measures of exercise capacity, microcirculation. We took blood for markers of oxidative stress and skeletal muscle biopsies in order to look at their epigenetic profile. So the characteristics, as I said, we had 12 males, and the reason that we went only with males was because we wanted to reduce the heterogeneity that you see between males and females, which was a particular interest to us because of the epigenetic work that we were doing. We wanted them to be all non-smoking, and we activity matched them for the Houston non-exercise rating. And for those of you who aren't familiar with that, it's a score that's self-reported between 0 and 7. And 0 is where you say that actually you avoid all exercise at all possible opportunities and don't do anything that might possibly stress you in any way. And if you're a 7, you're either walking 10 miles a week or doing at least 3 hours of exercise in a week, which might be playing soccer, it might be running, it might be swimming, it might be other kind of similarly related exercises. So quite a significant exercise profile. And the other thing that we asked for was that all of our participants had a VO2 max at sea level greater than 30 mils per kilogram per minute. And it was quite interesting. So when we went out to recruit the individuals, clearly the 6 who were altitude experienced were all professional climbers, because you don't really get anyone with that sort of altitude profile unless they're doing this all the time. But the altitude naive were local individuals who actually tended to be super obsessed with fitness and probably had higher VO2 maxes slightly than the altitude experience, although it wasn't statistically significant, as I will show you later. So this is the Ascent profile. As you can see, we did some baseline testing, and then at day 0, we took them up to the equivalent of 2,800 meters, which is where Monjo is in on the trek to base camp. And then they stayed at that equivalent altitude for 24 hours, and then they ascended up to 3,500 meters, which, as I said, is equivalent to the height of Namche, and they stayed there 48 hours, and then we brought them out. And this basically mimics the trek that we've taken all of our trekkers on over the course of the last 10 years or so with our Extreme Everest treks. And we have over 400 participants now that have followed this Ascent profile in the external environment. So the laser Doppler testing, we carried out the protocol at baseline here, and then after they had ascended to 3,500 meters on day 2, and then again 24 hours later at day 3, and then on day 4 when they returned to baseline. And we did the Doppler 1 hour post food, and also on those occasions where it was an issue on day 0 and day 3, we made sure that the laser Doppler was carried out before they actually did any exercise testing. So the protocol, you need to prepare the participant for this. So they need to be supine for 30 minutes prior. You need to tuck them up, make them comfortable, and make sure that they're in a position where they're not going to want to wriggle. You put the probes on the forearm and the index finger of the non-dominant arm. And with the forearm, you're wanting to make sure that the probe avoids large vessels, because that will affect the data capture. And it's important that you try and make sure that you always put the probe back on the same place. And so we used a permanent marker pen to draw around the probe position and made sure that they didn't scrub too hard in the shower over the various different days, so that we could make sure that we put the probe back on exactly the same place. We tried to keep the room temperature constant. We tried to minimize external stimulation, because any twitching then affects data capture. This is quite an issue. When you lie someone down and tuck them up nice and warm, they tend to fall asleep if you're not careful. You have to try and keep them awake without actually making them twitch. So the protocol itself included 7 minutes of data capture, where we did inspiratory breath holds at 3, 4, and 5 minutes, and then there's a 10-minute period of rest, and then after 10 minutes a blood pressure cuff is inflated on the non-dominant arm up to 300 millimeters of mercury. It's kept there for 3 minutes, and then it's released, and then we captured 5 more minutes of what we call postocclusive hyperemia data. And so the analysis plan was that we used t-test to look at data between groups, and then ANOVA within groups. So as you can see from the baseline characteristics, mean and standard deviation presented here, the groups looked very similar. There was no statistical difference in relation to any of these values at baseline. And over the course of the 4 days, there was no statistically significant difference in saturations between the 2 groups. There was no difference in hemoglobin levels, no difference in hematocrit, and no difference in estimated oxygen content. And the way that we calculated estimated oxygen content was that we multiplied hemoglobin by saturation by a constant of 1.34. So if we look at flux, which as I said before is measured in these perfusion units, this is altitude naive versus altitude experienced from day 0 to day 4. And if we look at the skin blood flux, we can see that between baseline and day 3, there is a significant reduction in skin blood flux in the altitude naive, but no real difference in the altitude experienced. And the thing that I find interesting about this data is that this group, there's much less scatter. They seem to be much more similar in their response. And then if we look at tissue oxygen saturation, again, we've got day 0 to day 4 in the altitude naive and the altitude experienced. You can see that between day 2 and baseline, and even more between day 3 and baseline, there is a reduction in tissue oxygen saturation in the altitude naive, but not the altitude experienced. And if we look at this post-occlusive reactive hyperemia, which is the vasodilation that you get after the occlusion, so you have this period of blood flow at rest, it reduces during occlusion, which is what you would expect, and then you get this big vasodilation after the blood pressure cuff is released, so you get an increase in blood flow. And you can see that in the altitude naive, that there is a significant reduction in that dilation after the occlusion at day 3 and day 4 in those that are altitude naive, but you see no significant difference between baseline and any of the days in those that are altitude experienced. So, in summary, there was no difference in the microcirculatory phenotype between the two groups at baseline. However, when we actually exposed them to the chamber to the hypoxia, the altitude experience demonstrated no real change, but the altitude naive demonstrated a reduction in both flux measured in perfusion units and tissue oxygenation. So the study numbers are very small. We have gone back to the chamber to do a validation study, and we will be looking to see whether a further 10 individuals demonstrate a similar picture or not. We don't know whether that difference is isolated or whether we would see it in greater numbers. And we don't know whether this is that those individuals that were altitude experienced went back to altitude again and again and again because they just deal with altitude better. So is it something to do with their DNA, or is it that their repeated return to altitude has changed their response to that hypoxic exposure? So I just need to say thank you to my collaborators, particularly the guys who ran the hyperbaric chamber for me, and my collaborators at UCL and the University of Southampton, and in particular the poor individuals who went into the chamber for 4 days and were allowed no alcohol, were not allowed to do a lot of exercise, which some of them found very difficult, and had to also have a low nitrogen diet because we were also looking at nitric oxide markers. And that for them, Americans like their bacon for breakfast. And of course, bacon is very rich in nitrate and nitrite. So they were quite upset that they weren't allowed caffeine either. I forgot to mention that. So quite a shame. Bless them.